Hello class, and welcome to this video, this short video that I'm going to explain the structure of the cell. So here we have the glass model of the cell. You could see the cell is made up of three distinct parts. This part over here represents the plasma membrane. This whole space in the middle is the cytoplasm, and this dense structure in the middle here is the nucleus. Within the cytoplasm, you have a lot of organelles that are around, and each one of these organelles have a distinct function. Let's start with the nucleus. The nucleus is separated from the cytoplasm by this white structure over here, which is the nuclear envelope. In that nuclear envelope, you could see those holes in here. These are called the nuclear pore. These are very important as a medium of communication with the cytoplasm. You see here a dense structure inside the nucleus. This is called the nucleolus. This nucleolus is the one responsible for making ribosome and ribosomal RNA. The space within the nucleus is called the nucleoplasm, and you could see those squiggly lines. These are representing the chromatin material. These are the condensed form of DNA found in a non-dividing cell. If you look at the cytoplasm had these structures in here. These represent the cytosol, the solution which is found within the cytoplasm. One of the first organelles we're going to discuss is this structure over here. This has those white spherical structure. These are the ribosome, and ribosomes are important in protein synthesis. Ribosomes are found in two different forms. Some of them are free, they are unbound to any organelle, and they are free ribosome. Some of these ribosomes are attached to the endoplasmic reticulum. The rough endoplasmic reticulum look rough under the microscope because of these ribosomes that are important in protein synthesis. Another form of ribosome, another form of endoplasmic reticulum are this one over here. These are called the smooth ER. You could see them over here. Smooth ER do not have the ribosome bound into them, and they are important in lipid and carbohydrate synthesis. So the rough ER are important in protein synthesis, and the smooth ER are important in lipid and carbohydrate synthesis. When molecules are made, either in rough ER or smooth ER, they are sent into this structure. This is called the Golgi apparatus. The Golgi apparatus has a receiving face, which is the one facing the endoplasmic reticulum, and it has a transport face. The receiving face is called the cis face, and the transport face is called the trans face. After packaging that molecule within the Golgi apparatus, you could see here they are sent out into the cytosol through secretory vesicles. These secretory vesicles contain various products that were made inside the cell so that we can transport them through the plasma membrane in the process of exocytosis outside the cell. In order to make protein through the rough ER, in order to make lipid and carbohydrate through the smooth ER, in order to package all of these different molecules in the secretory vesicles, we need energy. This is when this structure comes in place. This is the mitochondria. Mitochondria is also known as the powerhouse of the cell, and inside the mitochondria you could see those cristae, these folding, that are important in increasing the surface area of making energy in the form of ATP. So you could see here we have some of the storage vesicles that some cells would have. So some cells would make product and keep it inside the cell for later use, or sometime through those secretory vesicles we're going to excrete them out through the exocytosis. Now this structure over here, the creamy structure or yellow structures represented here and here, these are called lysosome. Lysosome contain digestive enzyme. These lysosomes are extensively found in white blood cells. White blood cells will engulf a bacterium and take that bacterium and merge it with the lysosome so that we can secrete those digestive enzymes on that bacteria and get rid of the body of that harmful pathogens. The second form of storage vesicles is this structure over here. You could see it here in gray. These are the peroxisome. Peroxisome contain oxidase, catalase, peroxidase. These are different enzymes that degrade toxic form of oxygen. So these are extensively found in the liver because the liver is the organ in the body responsible for detoxification of any harmful substances, including any form of toxic oxygen. Last organelle we're going to discuss are these two structures. These are called the centriole, all of these, and you could see they have their own amorphous protein around them. And these two centriole form something known as centrosome. So this structure material, the amorphous protein around them, and the two centriole form something known as centrosome. Centrosome are very important in making cytoskeleton. All of these organelles that you see in the cytoplasm are bound in place because of cytoskeleton that would maintain the shape of the cell, and also those cytoskeleton are important in cell division. So these two centriole are important in cell division and are important in making the cytoskeleton.